Okay, so we're on to part 2 of our study guide. This time we are looking at blood. So we'll be going through all the components of blood: plasma, platelets, erythroblastos fetalis, blood typing, etc. So we'll be going through all that and going through the answers to the study guide notes. And there's my cat for good luck. So we'll start with plasma. So components of plasma, there are proteins equal to 7% of plasma. And the reason I did this first is because I find that it's most important that we understand what these proteins are and what they do, because we're going to be asked these questions on the test. So starting with albumins, which main function is to maintain oncotic pressure and transport lipids, and they are derived from the liver. And I'll have you know that actually all of these are derived from the liver. So that should be pretty easy to remember. So your main function here is maintaining the oncotic pressure and then transporting lipids or fats is the way you can understand that. Globulins transport ions, hormones, and lipids, assisting in immune function made in the liver. So this mainly globulins are assisting in immune functions and also made in the liver. Fibrinogen, which is a big one that we will talk about in further detail, as you can see going down here. Fibrinogen is made in the liver and is factor 1 in forming a blood clot. So fibrinogen is the most important, I'd say, of the plasma components because it is used in the forming of a blood clot, and we'll go through that in further detail. Prothrombin will return into thrombin. Thrombin then catalyzes the transformation of fibrinogen into fibrin. So to explain what I just said there, prothrombin is the original protein that is found in plasma, but it will change into thrombin. And so then thrombin then catalyzes, which means it speeds up the transformation of fibrinogen, the other protein, into fibrin. And fibrin is what is used for the final form, essentially, for making the clot. And then so your prothrombin is also made in the liver. And so we'll go further into detail about this when we talk about blood clotting. But just understand that these are the proteins. So albumins transport of lipids, globulins assist in immune functioning and also transport of ions, hormones, and lipids. Fibrinogen, which is used for blood clotting and is factor 1. Prothrombin, which is also used for blood clotting, but it actually is turned into thrombin that then catalyzes fibrinogen to turn into fibrin. So the other component of plasma is water, which makes up 91% of plasma. So most of plasma is just made up of basic water. And then other solutes, which make up the other 2%. And so other solutes are mainly like your electrolytes and some gases that flow through the blood. Blood clotting factors. So this is what I said we would get more into. He only wants us to understand the last 2 steps of the blood clotting cascade. And so I'll explain those last 2 steps. And it's actually what I was just talking about up here. Prothrombin is a protein found in plasma, which I already knew that, and the liver, because that's where it's derived from, that changes into thrombin when being acted upon by factor X, or factor 10. So factor 10 is in the cascade, and towards the bottom, factor 10 will act upon prothrombin, changing it into thrombin. And so what you have to understand is factor 10 is just catalyzing the prothrombin transformation into thrombin. So you're changing this protein into this protein, okay, and by the use of factor 10 in the cascade. This is one of the steps we need to understand, is the change of prothrombin to thrombin. And then thrombin then acts upon another plasma protein called fibrinogen to create fibrin. And so this is the second to last step that we need to understand. So this is the first step, is the change from prothrombin to thrombin, acted upon by factor X. And the second step we need to understand is thrombin then acts upon another plasma protein called fibrinogen to create fibrin. Fibrin is the final form non-globular protein that then acts with platelets to form a plug or clot. Plug is just another term for clot. Essentially a plug to stop the bleeding, so a clot. Now I'll talk a little bit about platelets. Platelets are made in the red bone marrow, and their job is to clump and to form a clot. Okay, so this is more forming a clot. This process is called thrombopoiesis, or the creation of a thrombin, which is a blood clot. or the production of thrombocytes or platelets. So platelets and thrombocytes are actually the same thing. So thrombopoiesis is the creation of thrombocytes. So thrombocytes being just platelets, so clotting factors. The process is controlled by thrombopoietin, and the process begins with the stimulation of megakaryocytes, and these are found in the spleen and lungs. So megakaryocytes are your activating factor of thrombopoiesis. But the process is controlled by thrombopoietin. So megakaryocytes act upon thrombopoietin to start the process of thrombopoiesis, which will create thrombocytes. And I know that can get confusing with those three words being so similar. So what you need to understand is just break the words up into pieces. So thrombo is just stating thrombocytes, right? Thrombo meaning platelets. Poiesis meaning to give life. So thrombocytes, or thrombopoiesis, is the creation of, or the give life of platelets. And thrombopoietin is just what is controlled, that controls this entire scenario. And megakaryocytes is what acts upon the thrombopoietin to begin the stimulation and begin the process of thrombopoiesis to create thrombocytes. Okay? So that is plasma and your platelets and your blood clotting factors. There's a few more things I want to get into about that, though. There is calcium ions, which assist in the activation of thrombin and thrombins effect on fibrinogen. So calcium ions essentially work with factor X to create the activation of thrombin's effect on fibrinogen, changing it into fibrin. Fibrinolysis is the process of clot dissolution to remove clots in the body. So once we form a clot, it's good, it's helpful, it stops us from bleeding, but we don't want to keep that clot forever. Because if we do, that clot can go into our lungs, it can go into our heart, it can go into our brain. It can cause lots of problems later down the line. So our body needs to dissolve those clots so that we can continue to function without having to worry about that happening to us. And that process is called fibrinolysis. So the breakdown or the clot dissolution. And that's pretty much it you need to know. So there are multiple factors that work on blood clotting, mainly platelets, fibrin, and thrombin working on fibrin. And then also the calcium ions just kind of assisting in that process. And then fibrinolysis to dissolve or get rid of the clot after the process is finished and we no longer need it. So now we're going to talk about white blood cells. Okay, and there are 5 kinds of white blood cells, and those are also broken into 2 categories as well. So just talking about white blood cells in general for a moment, they make up 1% of whole blood. So of all whole blood, they actually only make up 1% of it. And that's because 55% of whole blood is made up by your plasma, which makes sense because it's mainly water. And then 45% is made up of your different parts that make up your red blood cells. And then the last 1% is made up of your white blood cells. And so it is also known as leukocytes. So white blood cells and leukocytes are actually the same thing. So they're interchangeable terms. So you have 2 categories. You have granulocytes, which are white blood cells with large granules. And there are 3 kinds of those. There are neutrophils, which are highly phagocytic cells capable of diapedus and also contain lysosomes. So lysosomes, as we remember, are digestive materials, digestive factors in the blood to get rid of waste. And so neutrophils are doing some of the heavy hitting of getting rid of things like infection. That's also why they're capable of diapedus, which is, for those who don't know what that word means, it means that these cells can, the neutrophils, can travel through the capillaries. So it can actually go through the capillaries to tissue. And that's how it can fight infection, is it can travel through those capillaries, through the walls, into the tissue, and then fight the infection. Eosinophils help regulate allergic reactions and protect against parasitic infection and release chemicals of immunity. So eosinophils, what you really want to take away from this, is that they're big players in chemicals of immunity. So they help with our immune system. Basophils contain histamine and heparin. Histamine is what causes allergic reactions. And then heparin is the thinning of our blood. And they are also capable of diabetes. Now, they don't do it as often as neutrophils, but they are able to do it. And then we have our agranulocytes, which do not have a stained cytoplasmic granules. So their cytoplasm is not stained. So our first type of that is lymphocytes, which are actually broken into two separate smaller sections as well. Lymphocytes are the smallest of the white blood cells, so they're actually the smallest cell out of all 5. And there are 2 kinds. There are T lymphocytes, which attack an infected or a cancerous cell. So again, big lymphocytes and neutrophils are your big heavy hitters in fighting your infections and also fighting cancerous cells as well. And there are B lymphocytes, which produce antibodies against specific antigens. And so antibodies working against specific antigens, with the immune system and fighting infection and fighting cancerous cells. And then we have monocytes, and they are the largest leukocytes. So they are the biggest white blood cell, because remember, leukocyte does mean white blood cell. They are highly mobile and highly phagocytic cells. So again, they're big removers of garbage that is in our system. And there are 2 things two terms really that we need to understand when we're about to take the test. There's leukopenia, which is a decrease in white blood cell count. And there is leukocytosis, which is an increase in white blood cell count. And so understanding that these 2 things can happen to the human body, typically leukocytosis happens when we have infection or something going in our body that our body requires fighting against it. And leukopenia, being a decrease in white blood cell count, is when someone is immunodeficient, right? Someone who has been getting chemotherapy may have a decreased white blood cell count. Okay, so we'll move on to the next section here, which is red blood cells lifespan and structure. So this is pretty important, and there is lots of things on the red blood cells, so we'll just go ahead and get into it. So their lifespan is 105 to 120 days. That's how long they will be in our system acting as red blood cells transporters. Macrophages use the process of phagocytose to remove old, abnormal, or damaged red blood cells. So that's after either the red blood cells have been damaged in some way, or perhaps they're were created improperly by our body, or they were damaged or old. So maybe they're past the 120-day mark, so they're not holding together appropriately, and so they're not transferring oxygen appropriately. And so our body will use macrophages to get those red blood cells that are old, abnormal, or damaged out of circulation so that we can have the most efficient red blood cells in circulation. And so hemoglobin is broken down, and amino acids, iron, and bilirubin are released. And I'll explain that a little bit more in detail later, but just understand that hemoglobin can be broken down into a heme ring and then globin. And then that globin is broken down into amino acids and iron, and the heme ring is broken down into bilirubin. And I'll talk more about that later. This is a structure of a red blood cell. There is no nucleus, and it is also a bioconcave disc. And so I'm sure all of you can picture a red blood cell in your mind, and that is what a bioconcave disc essentially looks like. They cannot duplicate themselves, so they don't go through the process of mitosis, and that's why they have this process of phagocytose, is so that old or damaged ones can be taken out of circulation and just brand new ones put in, rather than them duplicating themselves into 2 new cells. They also don't have a nucleus, so that's why they're unable to do mitosis. Their primary component is hemoglobin, and so that hemoglobin is actually what transports oxygen throughout the body. And I'll talk more about hemoglobin here shortly. Stimulus for making red blood cells is created by the kidney, known as and here's that word again, poietino. And so remember, this is the giving of life, right? Erythropoietin is poietin or poiesis are both meaning to give life. And so erythropoietin is the actor of erythropoiesis, which is the creation of red blood cells. This stimulus travels to the red bone marrow. So the erythropoietin travels to the red bone marrow, which begins erythrocyte production. Okay, and the process is called erythropoiesis. So again, you have that poiesis, right? The life. And so the only difference between this is this one's erythro. And as we know, red blood cells are also called erythrocytes. So erythrocytes are created by erythropoiesis. So to give life to erythrocytes. Okay. So I wanted to kind of break down the red blood cells lifestyle steps more clearly so that people can really understand the process they go through. So first, they're created in the red bone marrow by erythropoiesis, right? They have an 120-day lifespan going through our circulatory system before they start to break down. Once they break down, they'll be broken down into amino acids that are reabsorbed by the body, iron that is reabsorbed, and hemoglobin's heme ring will turn into bilirubin. And bilirubin will be excreted through feces. So we're actually going to poop that out. That's what gives pigment to feces, is actually the bilirubin from our blood. So red blood cells function is, you're actually looking at the function of the hemoglobin in the red blood cells. So hemoglobin is used for the transportation of oxygen throughout the body. So that's how we disperse oxygen all throughout our body, is it actually latches on to hemoglobin. And hemoglobin can attach 4 oxygens to make what's called oxyhemoglobin. And what that essentially is saying is hemoglobin by itself does not have oxygen in it. It's actually only in its oxygenized state when it's oxyhemoglobin. Otherwise, it's just hemoglobin, the main component of red blood cell. So hemoglobin, when it has been attached to 4 oxygen molecules, you'll have oxyhemoglobin, right? It makes sense. The combination of oxy being oxygen and hemoglobin being the red blood cell itself. Now, I want you to understand a couple of malfunctions that can happen with red blood cells. Kind of the same thing with white blood cells. So you have polycythemia, which is an increased hematocrit percentage. And for those who don't know what hematocrit is the percentage of whole blood that is red blood cells. And the average range for that is 45%. And then for women, I believe you can actually go down to 42%. But so hematocrit is just the percentage of whole blood that red blood cells occupy. Okay. And so anemia is a lower hematocrit percentage than normal. And so anemia, can typically happen again in people who are immunocompromised, and it's also a disease we just kind of see commonly at the hospital. Okay, I think this next chart is going to be very helpful for people who are having a hard time with blood compatibility. I made this chart so that people can see exactly what pairs with what. So we'll start with describing the types. So we have type A, blood, and it has antigens to A, meaning it is compatible to A, right? And antibodies to B, meaning it is not compatible to B. And then type B is the reverse of that. It has antigens to B, so that it is compatible with B. And then antibodies to A, so it is not compatible with A blood. And then we have type AB, which has antigens to both A and B, and antibodies to nothing. So it can accept actually all kinds of blood. And we'll kind of get into that a little bit, because AB negative technically can accept everything, but AB positive actually can accept every single type of blood. It's actually the universal acceptor. And then we have type O. And so type O has antigens to nothing. It is not compatible with A or B. It cannot receive A or B blood, but it has antibodies to A and B. So it cannot receive those bloods, but actually it can donate to those kinds. And we'll get into that when we talk about our chart right down here. So looking at our chart, we have our donors across the top going from O negative to AB positive. And then we have our recipients down here. So we have O negative to AB positive. And we did a kind of little cross section here. So O negative, as you can see, can donate to every single kind of blood type. And that is why it is actually the universal donor. So O negative is your universal donor. And AB positive can accept all kinds of blood from everyone. And that is why it's your universal acceptor. Now, I want you to understand this. O negative can donate to O positive, but O positive cannot donate to O negative. And so we think, why is that? And the reason for that is there is a written rule that negative types of a blood type can donate to their positive friends, but positive friends cannot donate to their negative type. And so let me give you another example of that. A negative can donate to A positive. So A positive people can accept A negative blood. However, A positive people cannot donate to A negative blood because of the antigens. So Rh negative people, meaning they have a negative symbol next to their blood type. So if you're A negative, B negative, O negative, AB negative, and if you're negative at all, you have the Rh negative antigen, which means you have the Rh positive antibody. So if you're negative blood type, you cannot accept your positive counterpart. However, if you are a positive Rh antigen, then you can accept your negative partner. So A positive can accept A negative, but A negative cannot accept A positive. And that works for every single blood type. O positive can accept O negative. O negative cannot accept O positive. And like I said, that works for every single type. O's, A's, B's, AB's. It's all the same. So I hope that this chart can help you, and I hope that that little rule can help, that negatives cannot accept positives, but positives can accept negatives. And as another general rule, just so we understand we're on the same page, A's can only accept A's and O's. O negative, more specifically. But A's cannot accept B's. They cannot accept AB's because AB's have a B in them. And so you have to understand what you have to look at is A's can only accept other A's and only in specific conditions when it is a negative not accepting a positive, but a positive accepting a negative. And so just remember my earlier little tip for you that look at your antigens and your antibodies, but then also look at what kind of blood you're trying to start with. So if you're starting with your A negative, don't accept your A positive. But if you're starting with your A positive, then you may accept your A negative. So just understand and know that rule. The last thing I want to discuss is erythroblastosis fetalis. And so this kind of goes with the Rh antigen antibody thing I was talking about. So if you have an Rh negative mother, okay, so the mother has negative blood. It could be any kind. We could say it's A negative. Okay, so the mother has A negative blood. Can kill an Rh positive child. And the reason for that is because if the child has A positive blood, okay, and the mother's system that cannot accept A positive blood finds out that there's this child on the inside of her, okay, that's basically full of A positive blood, the body is going to react by trying to eliminate that A positive blood. Okay, so what happens is if you have a mixture of their blood somehow, so let's say something happens that destroys the barrier between the child and the mother's blood, so they mix. The antibodies that are in this Rh negative blood are going to hunt and kill any Rh positive blood that they find. And what can happen is the mother's immune system could end up going to try to kill all of the blood cells in the child once they mix. And so if that happens, the child can effectively have his blood or her blood destroyed until the point of the child dying. And so that's what erythroblastosis fetalis is when the mother's immune system attacks the child inside of her body because they have opposing positive and negative charges on their blood type, but it is only when the mother has a negative blood type and the child has a positive blood type. If it is the reverse, so if the mother has a positive blood type and the child has a negative blood type, this won't actually occur because the positive blood type can accept the negative blood type. And so that's it. That's all of our blood notes. If you have any questions or need something reviewed, please let me know. Thanks.